we're talking about today is protein translocation, which is the process by which peptides are transported across the membrane bilayer. Translocation of proteins across the membrane of the endoplasmic reticulum is known to occur in one of two ways: co-transitionally, in which translocation is concurrent with peptide synthesis by the ribosome, or post-translationally, in which the protein is first synthesized in the cytosol and later is transported into the ER. Both means of translocation are mediated by the same protein channel known as SEC61 in eukaryotes.
We will be focusing on post-translocation process, which begins in the systole because of the fact that all protein synthesis in the eukaryotic cell begins in the systole. This is where ribosomal subunits come together to begin translating an mRNA near the 5' end. Begins to leave the ribosomes. They are then moved toward the endoplasmic reticulum. If made to be included in the ER, it's made to be included in the ER Golgi apparatus lysosomes or plasma membrane. The protein enters the ER co-translationally, which means that the proteins that are entering the ER have a signal sequence in the amino terminus, which can be located in the polypeptide chain, and the signal sequence spans 20 amino acids and has hydrophobic amino acids. The signal sequence emerges from the ribosomes, recognized and bound by the signal sequence, which is the SRP recognition particle. ER binds to the SRP receptors, and as signal sequence proceeds, the signal sequence is then cleaved by the signal peptides and then the polypeptide chains through the translocation of the ER.
N-terminus is represented in orange, and it is a cluster of about 8 hydrophobic amino acids at the N-terminal end of a protein. This sequence remains in the membrane and is cleaved off of the protein after transfer through the membrane. The C-terminus is represented in blue, and it is the end of an amino acid chain terminated by a free carboxyl group.
The ER start signal is shown in green. The signal of the ER entry is 8 or more hydrophobic amino acids and acid residues, which rivets the polypeptide into the ER membrane and is also involved in translocation. The ER stop signal is shown in red, and it is located at the N-terminus and is cleaved by the protein in the rough ER lumen.
The Cadel sequence, represented in yellow, prevents a protein from being secreted from the endoplasmic reticulum and facilitates its return if it is accidentally exported. The purple KKX signal is a signal sequence located in the cytoplasm for transmembrane translocation.
The nuclear localization signal is shown in yellow and is a sequence of NLS is shown an amino acid sequence that tags a protein for import into the cell nucleus by nuclear transport, and the mitochondrial targeting signal is shown in black. It is 10 to 70 amino acids long peptide that directs a newly synthesized protein to the mitochondria. It is found at the N-terminus and consists of an alternating pattern of hydrophobic and positively charged amino acids to form what is known as amphibic helix.
Polypeptide set A. Polypeptide sequence 1 with orange N-terminal goes to the mitochondria. Then with blue C-terminal, it stays in the mitochondria. Sequence 2, orange N-terminal with green ER start signal. So it enters the ER, then leave and stay in the cell. Sequence 3, orange N-terminal with green ER start signal. It enters the ER, then red ER stop signal. So the sequence stays in the ER. Sequence 4, orange N-terminal with green ER start signal, then red ER stop signal. So it stays in the ER. Then another green ER start signals of the same sequence enter the ER, then red ER stop signals, so the sequence stay in the ER. Sequence 5, orange N-terminal with black mitochondria signal go to the mitochondria, then yellow nuclear location signal, so the sequence go to the nucleus, then green ER start signal, so it enters the ER, then leave the ER and stay in the polypeptide sequence 1, orange N-terminal with yellow KDEL signal to do retention and retrieval, so it enters the ER, then go out, then come back and stay in the ER. Sequence 2, orange N-terminal with green ER start signal, so it enters the ER with yellow KDEL signal. It leaves and then come back to stay in the ER. Sequence 3, orange N-terminal with green ER start signal, so it enters the ER, then purple K-triple signal, only do retrieval, so it leaves and then come back. Then with red ER stop signal, the sequence stays in the ER. Sequence 4, orange N-terminal with green ER start signal, so it enters the ER. Then with red ER stop signal, the sequence stay in the ER. Then purple K-triple X signal, so it leaves, then come back. then leave the ER again to stay in the cell. Sequence 5, orange end terminal with green ER start signal. Then with red ER stop signal, the sequence stays in the ER. Then purple K triple X signal, so it leaves, then comes back, then leaves the ER again, then with another green ER start signal, then red ER stop signal, the sequence stays in the ER. Sequence 6, orange end terminal with green ER start signal, then red ER stop signal, so the sequence stays in the ER. Then another green ER start signal, so it enters the ER with yellow KDEL signal. It leaves, then come back to stay in the ER. Sequence 7, orange end terminal with green ER start signal, then red ER stop signal, so the sequence stays in the ER. Then another green ER start signal with red ER stop signal, so the sequence stay in the ER. Then with yellow KDEL signal, it leaves, then come back to stay in the